Okay, so my name is Rhea Agarwal, and I'm presenting my research on vaccine designing for the Lassa fever virus using AI and computational tools. And I was mentored by my PhD mentor Pallavi Agarwal. So I would like to first start off with my objective, which was designing a multi-epitope vaccine against the Lassa fever virus using computational methods. And through the research, I basically evaluated the vaccine construct using its physiochemical properties and its potential to trigger immune responses. So I used AI driven computational tools to first find out the proteins, analyze the viral proteins, assemble epitopes, and then model and refine the structure using simulations and molecular docking. And this is very significant, it's very impactful since there is an urgent need to prevent the Lassa fever virus, as it's a very neglected but very rampant disease. And this research basically proposes a promising vaccine candidate, which can also be used for further experimental validation since I have not done a wet lab testing. I've also demonstrated through this research a rapid in silico approach to vaccine development. So, as I said, right now, Lassa fever virus is a very significant public health threat, and it's a neglected disease. So, it doesn't have any licensed vaccine. It is very easily transmitted, and it has long-term health impacts. And for this, to alleviate this problem, I have researched on possible vaccine epitope types. And this multi epitope approach basically um, makes sure that any genetically enhanced or variable version of Lassa fever virus is also inhibited, and it can also have a more aid to a more robust immune response. The vaccine is also made such that it can also be adjusted, and it is very easy and safe to use since the vaccine itself doesn't pose any health threats, and it is also looked into. If more research is done, then actual units can be produced. So basically what my objectives were, first select the epitopes, and then evaluate the efficacy, and then use the computational and AI tools to enhance the design and evaluate the performance of the vaccine. This is some of the literature review that I did beforehand, and also the softwares that I use. So I used computational software such as Vaxigen, and I also made sure that I research what the current scene was with the Lassa fever virus, which is that they have discovered that the virus is very potent and very it's a huge problem, but actual vaccines have been only recently discovered, and they are still in development and in testing. So I have research upon hopefully is so as I discussed before that this is my methodology. I used computational tools such as Vaxigen, and then I designed the construct. So basically the epitopes which I found out through the computational tools, I assembled them for immunogenicity. Then using simulations, I assessed all of the epitopes and their connection with each other, and then in vitro testing was done to evaluate immune response, antibody production, basically make sure that the vaccine is actually working. This is some of the analysis and development that I did. So target antigen selection, basically I chose a GPC which was processed into surface glycoproteins, and this made sure that it could penetrate the host cell, which was the vaccine cell, better. And epitope prediction and screening was done through a computational and AI tools. And then the actual vaccine construct design was also done through computational tools, and it basically included all of the beta defense 3 adjuvants, chemical chemicals, and basically then I checked it for immunogenicity. After that, I also used a software called Protopram to test its chemical and molecular elements, and then I made sure that I was checking it for antigenicity, non-allergenicity, to make sure it's actually usable. Then I used structure modeling softwares such as Swiss Model and Haddock platform to basically evaluate its interaction with the disease. Then finally, I used a Monte Carlo simulation for actual immune response prediction. After I obtain the results, I analyze them that several B cells, CTL and HTL epitopes from the Lassa virus proteins. I was able to find highly antigen and non-allergenic epitopes, and Vaxigen scores I received were over 0.4, which indicated strong antigenic potential. And the vaccine itself consists of multi-epitopes, which includes beta defense adjuvant, CTL, HTL, and B cell epitopes. And the construct itself is 129 amino acids long, and it has a high Vaxigen antigenicity score, which is around 0.45, and it is confirmed to be non-allergenic. As I discussed earlier, this is a very potent and a very large health threat, especially in areas of West Africa. And using AI, computational tools, and immunoinformatics, I was able to design a vaccine which can be significantly streamlined, and it can really help to alleviate this problem. And right now, the testing is mainly for this virus done on animals, and therefore we know that it is very it spreads through rodents. To control Lassa fever virus, it's also imperative that we have such awareness to make sure that it doesn't happen in ways other than the vaccine. And there are some limitations as well with this vaccine, that it lacks empirical validation, it lacks wet lab testing, and it also doesn't account for any host variability, which is something that I hope that I can work on in the future. As for the actual vaccine that I have developed, um, it has demonstrated a strong ability against disease, and it has increased antibody titers. It is it has huge potential to reduce infection rates and improve health, safety, and outcomes in specific areas. It also has a non-replicating nature and at any adverse side effects, which makes it very ideal for use. And further research on it, further optimization can enhance efficacy of the epitopes, and it can also be enhanced for any other variability in the disease which is not considered in this research. That is the end of my presentation. So I had a really positive experience with the program. So I really enjoyed working with my mentor, and it was really beneficial. I learned a lot of things. Like the project that I worked on is not really something I would have learned in school, and it is really good for my education and that I did this.